So today is our topic is from the BRS physiology, chapter 1, cell physiology. It first we start from the cell membrane are composed of primary phospholipid and proteins, lipid bilayer. The phospholipid and glycerol have the backbone which is hydrophobic. Backbone is made up of the hydrophilic water soluble and head which is called head and two fatty acid layer which are the hydrophobic water insoluble. The hydrophobic faces each other with a form of lipid bilayer. Okay, then we have the lipid soluble substances like oxygen, carbon dioxide, and steroid crosses cell membrane because they cannot dissolve in the hydrophobic lipid bilayer. Okay, then we have the water soluble substances. Sodium chloride, glucose, and water cannot dissolve in the lipid of the membrane but may cross from the water filled channels or pores or may be transported by the carriers because they are insoluble in the lipid, so they have to transport through the water filled channels or pores or maybe from the carriers. Then we have the proteins. Proteins have the integral proteins and the peripheral proteins. Proteins, integral protein embedded in the cell membrane through hydrophobic interaction. Where are hydrophobic interaction? There are embedded proteins may span the cell membrane, include ion channel transport proteins. So we have proteins which carry which transpose the substance other than fat soluble. Okay, these are embedded in the cell membrane like transport protein, receptor, guanosine triphosphate, or binding protein. These all are made of the proteins. Peripheral protein are not embedded in the cell membrane, are not covalent bond to the membrane. Attached, that they are usually attached to the electrostatic interaction only. Okay, so we have okay, so then we have intracellular connections. So in intracellular connections, tight junction, your intracellular connection, other is intracellular connection. These are the tight junctions. So zonal occludents are the attachments between the cell. Often the epithelial cells may be in intracellular pathways for the solution depending on the size, charges, and characteristics of the tight junction. Okay, these are maybe tight impermeable as the renal distal tubule or leaky permeable as in the renal proximal tubule and the gallbladder. So tight junction may be tight or in the leaky, tight or the impermeable as in the renal distal tubule. There no any chance of the leaking, but other are leaking. That are leaking means they are permeable as like renal proximal tubule, PCT, and the gallbladder. Other intracellular connection or gap junction are the attachment between the cell that permit the intracellular communications. For example, permit the flow and electric coupling between the myocardial cells. A gap junction between cell and intracellular communication, it means the flow of electric coupling between myocardial cells. Permit the current flow electric coupling between the myocardial. Then so this is our all about the cell membrane. It has a lipid bilayer, a protein, and the intracellular connections. Lipid bilayer are the water insoluble but fat soluble. They can cross the lipid soluble substances, but for lipid insoluble substance we require the channel proteins or the water filled proteins. They are integral protein, peripheral protein. In this some integral protein embedded in the cell membrane, but peripheral protein cannot are not embedded in the cell membrane. So other one is intracellular connection in between the cells. It inter means in between the cell and other characteristics. Tight junction and gap junctions are the attachment to cell epithelium. Zona tight junctions are called zonula occludents, and gap junctions are the coupling between the myocardial cell. So, that's the example. Okay, then we go to room the transport through the cell membrane. We have a table like how the transportation of cell occurs. Okay, first of all we have the simple diffusion, facilitated diffusion, primary active transport, co-transport, and counter co-transport. Okay, a simple diffusion, it means electrochemical gradient. Electrochemical gradient means something Electrochemical gradient means something coming downhill or uphill. It means simple diffusion, which is from a higher concentration to the lower concentration. It can flow like we spill water from a higher higher status to the lower status. The water can easily slip down, so we don't need any carrier or any energy. So it simple diffusion is like that. So we don't any need carrier or any metabolic energy, simple diffusion. That's why it is called as simple diffusion. Facilitated diffusion. What is facilitated diffusion? Facilitated diffusion, it is a type of diffusion, but it is facilitated by some kind of protein. It is also a downhill procedure, but in some circumstances we need a carrier mediated that for diffusion, that is called facilitated. The carrier protein facilitate that substance to pass out from from a higher to lower concentration. Okay. And then we have a primary active transport. It is uphill. It means we are going from down to upward position. So we need energy, and we also need protein and carrier protein or carrier mediators. And these are the inhibition of potassium and sodium potassium pump is the example of the active primary active transport. Then we have a co-transport. Co-transport is uphill. It means we are going from lower concentration to the higher concentration. It also need a carrier mediate. It also need an indirect metabolic energy. Co-transport mean co-transport mean something is coming with something. Okay, matlab someone yes, some same direction inhibits by the abolishing of sodium gradient. Okay. Counter co-transport is it also a uphill transportation. It also need a carrier transport. Metabolic energy uses but indirect because it's counter, yes, opposite direction. Sodium gradient is needed when sodium passes in co-transports. When sodium passes, like glucose and other substance may pass with the sodium. In counter co-transport, when sodium passes but something going out, when sodium going in, the other substance going out. If sodium going out, other substance going in, inhibit by abolishing the sodium gradient. Okay. And then we have a simple diffusion. That's only form of transport that is not carrier mediate, occurs down electrochemical gradient, as I told you first, and does not require metabolic energy, therefore is passive. Diffusion can be measured using the following equation. Okay. JPAC1 minus C2, where J is a flux flow. We measure in the flow permeability centimeters and squares. A area square. C1 concentration and C2 the concentration. Okay, sample, it's like something mathematical. So we will you can do it's biologically, and I will tell you in another lecture. It will be going permeability. It is the P equation. Describe the ease with the E solute passes through the membrane. Radius of increase the diffusion coefficient, speed of diffusion. Radius of the solute increase the diffusion coefficient, speed of thickness decreases the diffusion distance. Okay. Small hydrophobic solutes like oxygen, carbon dioxide, the highest probability in the lipid membrane. Okay, hydrophilic solution, sodium, potassium must cross the cell membrane through the water-filled channel. Pores, hydrophilic. If a solute is an ion charge, their flux will be dependent on carrier mediated transport. Include facilitated diffusion, primary and secondary active transport. Characteristic of the carrier mediated stereo specificity. For example, D-glucose, a natural isomer is the transport by the facilitated, but L isomer not simple diffusion. The stereo specificity, for example, D-glucose is transport through the facilitated diffusion, but the L isomer is not transport through the diffusion. A convenient distinguish between the two isomers. Okay. Saturation, the transport rate increase, the concentration of solute increase until they carry the saturated. The transport maximum is analogous to the maximum velocity is enzymatic kinetic competition. Structure related to a solid transport the carrier molecule. For example, galactose is a competitive inhibitor of glucose transport. Okay, okay, we are stopping there. Then we start from the facilitated diffusion. Thank you.